Greetings everyone, my name is AKMAL bin ROZAMI. So today my group has prepared a video presentation and our topic is production of secondary metabolites in vitro. So these are the members of our group. I am AKMAL and then we have NUQMAN, AZIZUL, ALYSA and IMAN. The first part of this presentation is the overview covering the definition, major classes, roles and function in plant and pharmaceuticals. Secondary metabolites can be defined as organic compounds produced by an organism but play no role in its normal development. Secondary metabolites frequently play a crucial role in the plant defense, but they are not essential for the immediate survival of the plant. So there are four major classes of secondary metabolites in plant. The first one is alkaloids, a nitrogen-containing basic compounds, and they aid defense against herbivores and pathogens because they have toxicity property. Some examples are morphine, caffeine, and nicotine. The second major class is flavonoids. They are polyphenolic pigments. They function as antioxidants and UV protectants, also serve as signaling molecules and antimicrobial substances. Flavonoids also attract pollinators through color and fragrances. Some examples are quercetin and anthocyanins. The third major class is terpenoids derived from isoprene. They provide defense against herbivores and pathogens and also attract pollinators and beneficial insects. They can also serve as signaling molecules in plant-to-plant and plant-to-insect interactions. They are also involved in plant growth regulation and environmental adaptation. Some examples are pyridine, gossypol, and taxol. The fourth major class is phenolics, aromatic rings with hydroxyl groups. So phenolics provide structural support. They can also act as antioxidants and UV screens, and they serve as attractants for pollinators. Phenolics also have ability to inhibit pathogen growth and aid in wound healing. Some examples are lignin, tannins, and salicylic acid. In vitro plant tissue culture methods are employed for secondary metabolite production. So what are the reasons why we choose in vitro methods? So the first reason is control and optimize production. In vitro systems eliminate any environment variability, so this offers environmental independence, and it also allows stresses such as light and temperature to be controlled optimally. The second reason is scalability and efficiency. So in vitro methods allow large-scale production in bioreactors, so this can help us achieve more yields, and these methods also accelerate biomass production. The third reason is purity and consistency. Contamination can be prevented through in vitro methods, and it also ensures batches of metabolites produced to be standardized. And the fourth reason is access to rare compounds. So methods such as hairy root culture can express metabolites more than normal roots planted in field without harming them. And also colorless induction can produce compounds absent in mature plants.
Next, let's take a look to the methods for enhancing the secondary metabolites. This method is electrocutation. So this technique involves exposing cells or tissues to certain stimuli, which is elicitors, and this is to trigger the biosynthesis of secondary metabolites. So elicitors mimic stress conditions, prompting the organism to boost its natural defense mechanisms, which often includes metabolite production. So basically, there are two types of it, which is biotic elicitors and abiotic elicitors. So for example, applying UV light to plant cell cultures can significantly increase the production of flavonoids or alcoholites. It's a cost-effective and non-genetic method widely used in labs and industry. The second one is precursor feeding. So this method involves adding metabolic precursors. So it is essentially raw materials into the growth medium lah. So the idea is to supply the biosynthetic pathway with more substrate to push the system lower increases metabolite output. For example, adding phenylalanine and tyrosine to a culture medium can boost the production of flavonoids or alkaloids. This is especially useful in controlled environments like plant cell cultures or microbial fermentation systems. The third one is genetic engineering. So this involves modifying the organism's DNA to either increase the expression of a key biosynthetic genes or to introduce new genes that improve metabolic synthesis. So in this case, we can use tools like CRISPR, agrobacterium-mediated transformation, and RNA interference. So these methods allow scientists to create genetically modified organisms that consistently produce higher yields of targeted secondary metabolites. For instance, inserting genes responsible for a cancer drug production in a microbial host has helped reduce reliance and endanger new trees. The fourth one is metabolic engineering lah. So instead of altering just one gene, metabolic engineering involves modifying entire metabolic pathway. It uses system biology, computational models, and synthetic biology to redesign how an organism processes nutrients and build molecules. For example, scientists can redirect carbon flux within a microbial cell to favor the production of a specific antibiotic while reducing the production of unnecessary byproducts. It's a complex but powerful approach, ideal for industrial application lah.
So let's move on to the next part. one of the key strategies is biotransformation. This process converts simple molecule into complex secondary metabolite using enzyme or precursors. It's commonly done using systems like hairy root culture, cell suspension, or microbial platform. Biotransformation not only increases yield but also improves structural diversity of the compound produced. For example, the conversion of geraniol to citronellol is a classic case, resulting in more valuable metabolite through enzymatic action. Next, we have scale up using bioreactor, which is essential for commercial level production. This involves cultivating plant cell or tissue in large, sterile, and controlled environment. Systems like cell suspension culture, hairy roots, or organ culture can be scaled in bioreactors such as stir tank reactor, air lift reactor, or wave bioreactor. This allows consistent and high volume production of target metabolite, which is critical for pharmaceutical or industrial application. Moving on to media optimization, this focuses on adjusting the culture medium to maximize both growth and metabolic synthesis. This includes tweaking nutrient level, especially nitrogen and phosphate, choosing the right carbon sources like sucrose, and adding natural booster such as coconut water. pH also play a key role. Usually a slightly acidic range around 5.6 to 5.8 is ideal for enzyme activity and cell metabolism. Finally, plant growth regulator or PGRs are vital in directing how plant tissue grow and what they produce. ALZIM and cytokinins are used to control root, shoot, and callus formation. Meanwhile, jasmonates and salicylic acid act as elicitor to enhance secondary metabolite synthesis. The type and concentration of PGR need to be customized depending on the plant species and the specific stage of culture.
Now, I, ALYSA, will be presenting the case studies, which are examples of successful in vitro secondary metabolite production. Let's look at the first example, which is Withania somnifera, or Indian ginseng. In this, hairy root cultures were successfully induced using Agrobacterium rhizogenes. The transformed root grew rapidly in MS medium, supplemented with 40 grams per liter sucrose. Importantly, they produce up to 2.7 times more than withanolide compared to the non-transformed roots. This highlights the effectiveness of this. The next example is Thymus vulgaris, or thyme. It was cultured both directly and indirectly using shoot tips or callus induced with 2,4-D and kinetin. The bioreactor systems with high carbon dioxide levels and elicitors like PEG led to a notable increase in essential oils production such as thymol, carvacrol, and rosmarinic acid. These in vitro cultures offered better essential oils consistency and quality compared to wild plants, hence making them ideal for commercial production. Lastly, in Angelica glauca, which is a Himalayan herb, the rhizome buds were cultured on MS medium with benzyladenine and gibberellic acid, achieving 94% shoot regeneration. The callus was induced from roots using 2,4-D and kinetin under dark conditions, and then they were transferred to liquid suspension cultures. The use of paraffin oil in the medium actually helped trap the volatile compounds. The GCMS analysis detected over 206 compounds including ligustylide and rhodopin. When treated with methyl jasmonate or sucrose, which is the elicitor, the culture showed increased monoterpenes and fatty acids, highlighting their potentials for producing high-value bioactives.
So we go for the next part, which is the application and the challenges in vitro production of secondary metabolites. So we go for the application, we can apply all of this in pharmaceutical. For example, the paprika cell from the Texas cell culture, so only large-scale commercial with success. Other than that, we can apply these in nutraceuticals, cosmetic and agrochemicals. So flavonoids, terpenes, and alkaloids is the example that we use in supplements, skincare, perfume, and pesticides. So we can enable the consistent year-round production regardless of the season. So other than that, we can apply this in sustainable and ethical production. We can use the wild plant harvesting, which is we can protect our biodiversity. We also can grow this in sterile condition, means no pesticide and herbicides. And lastly, we can apply this in metabolic engineering and microbiofactories. We can enhance yield using elicitors, precursors, and gene editing. Microbes such as yeast and bacteria now engineered to produce plant compounds more efficiently. So we also have to face a few challenges, for example, the low commercial scalability, because only a few compounds are commercially viable. Other than that, we have the high operating costs, because of the expensive lab setup and the labor, which almost all process not cost effective compared to the field grown plants or chemical synthesis. We also have the biological limitation, which is the low yields due to the unstable or incomplete biosynthesis pathway, which can lead to the risk of contamination and genetic instability over the time. So we have the regulatory and market barrier. For the example, GM-based product mostly face the strict approval and consumer skepticism, and the process optimization is complex because it requires a fine-tuning of media, hormones, elicitor, and genes.